So in our notes, typically what I have is some blanks, some small things for you to fill in as I'm talking, as we see on the screen. Usually it's not a ton, but I feel like it helps you to follow along and make sure we're sort of engaged in listening. So we're talking about scientific inquiry. Inquiry is a word. What does it mean to inquire?
Like the wonder, question.
Kind of to wonder. Yeah, inquiry is like asking questions. You're inquiring about something. It means to ask questions, to try to learn about something new. And so that's what really science is. Science is about a way of learning new things about the natural world. Sometimes we think of science like just a bunch of facts. Like this book, it's a biology textbook, and it has a whole bunch of facts and information that scientists have learned over the last several hundred years. So that's one way to think about science, like just a bunch of stuff that you can memorize or read about. But really what science is, in actuality, it's really a way of learning more things about the world. That's really what science is. It's a way of thinking. It's a way of asking questions and then finding ways to try to answer those questions. Experimenting, observing the natural world to help build our knowledge about the natural world. So it's not just what's in this textbook. It's really a way of thinking about the world. It's a way of approaching learning new information. And that's what we were doing yesterday. What we were doing yesterday wasn't really so much about like learning some facts about seeds. It was about asking questions about those seeds and germination and plant growth, and then trying to figure out a way to answer those questions. That's what we did yesterday. And that's what science is. So we call that a process. It's a process that we can use to answer questions about the world.
Okay.
Anybody know any of those scientists on the slide?
Well, I think I know some.
You know one, Luke?
I know all.
Yeah, I think I know them all.
You know all of them?
I think that's Marie Curie, James Goodall, Stephen Hawking, and Neil deGrasse Tyson.
This is actually I think this is Rosalind Franklin, but I'm not sure. Who did you say, Marie Curie? I don't think it's Marie Curie. Well, good job.
Oh, thank you.
Yeah, Jane Goodall worked in Africa studying the great apes.
Wait, what was that first two?
The first one?
Yeah.
I think it's Rosalind Franklin who.
Okay, Rosalind Franklin. Let's write it down.
Yeah, Rosalind Franklin. We'll talk a little bit more about her.
What about the second person?
Hold on one sec. Because Rosalind Franklin, she was important in the discovery of DNA. There's an interesting story about her. Is basically the two scientists that really won the Nobel Prize for understanding DNA were Watson and Crick. Rosalind Franklin actually was a woman who worked at the same university as them. She took these pictures of a DNA molecule using X-ray crystallography, and somebody kind of saw her pictures that she did, and that then gave them to Watson and Crick, who then, like, her pictures actually prompted us to understand the structure of DNA better. She didn't win the Nobel Prize. She didn't get credit. They almost kind of stole her work. So it's kind of an interesting story about that. Jane Goodall studied the great apes in Africa, famous scientist. Stephen Hawking, what was he? What was his type of science?
What, like space?
Yeah, physics.
I also know Mike deGrasse Tyson as being that flat Earth conspiracy theorist.
Oh, really? So Neil deGrasse Tyson, again, another famous scientist.
Yes,
I think astrophysicist. Somebody that studies physics and sort of astronomy. He does a lot of like public communication too regarding science. So that's why
I see planets around him.
Yeah.
What's an astronomist?
Somebody studies planets.
Yeah.
So let's talk a little bit about like how science works. So if you remember maybe back in junior high, maybe you learned about like the scientific method, that there's these set of steps that scientists follow, and this is how an experiment is set up. I mean, the reality is there's lots of different ways that science happens. We kind of simplify things sometimes in school and to a more manageable, but there's lots of ways scientists learn new information. They can take lots of different pathways to get there. There really isn't one single scientific method. This is one kind of version of a way that we can use science to learn more about the natural world. And so often we start with questions. And that's what we did yesterday. Right? I asked you in your groups to come up with questions that we could ask about these seeds and plant growth. So that's lots of times the first question. Scientists are always looking around the world, observing things, and trying to ask interesting questions, questions that can help us advance what we know about science. And then, after a question has been proposed, the next step usually is to make a hypothesis. A hypothesis, you did that yesterday as well. It's kind of like what you think. So you ask a question, and scientists actually answer the question before they set up an experiment. Now, they use their best information to come up with what they think the answer is, but they don't know if they're right or not until the process is continued. So we make a prediction. That's what a hypothesis is. It's a prediction about the question we're asking. So yesterday we set up, let's use the light experiment. We have these seeds germinating under artificial light versus natural light. And then you came up with some hypotheses about that. What did you guys think? What was your hypothesis, right?
I think we did that plants or seeds would grow better under sunlight.
Okay. So that's like that's your hypothesis. It's your prediction. Ray said he thinks they'll grow better under natural light than artificial light. We don't know yet. We're in a process of finding out. But that's coming up with a hypothesis is an important part about how we do science. And what we do is we test that hypothesis with an experiment. Okay? And so an experiment tests what we thought and sees it and tries to determine is it correct or not. So that's where we are in our process right now. We set up our experiment.
I understood all of this stuff back in
Good.
7th grade.
So our experiment's going on, and we are at step number 4, sort of. We're eventually going to collect some data. We're going to look at the results. We're going to see how many seeds germinated, or how quickly how fast, how much they grew. And then our next step will be, okay, to make a conclusion, to determine, okay, Ray thought the seeds were going to grow better, germinate better in natural light. We look at our data and we say, was that right or not? Was that correct? And we come up with a conclusion. So yes, my hypothesis was supported by this experiment, or maybe it wasn't. It's okay either way. Sometimes our hypothesis was not right. We still learn something from that, which can lead us to new questions or new hypotheses. Sometimes our hypothesis is supported. Okay, and we've, again, learned something from it. So this is a way of thinking about how we learn about the world. So when we talk about scientific inquiry, the scientific method, we're talking about how do we get new knowledge using science? How do we learn new things? So we start with questions. But not just any question, right? So science can't answer the question of what is my meaning in life?
What should I write down for what is science?
What's that? And so we have to answer questions that an experiment can actually help us answer. So good scientific questions are typically something we could answer through experimentation. Like the questions that you posed yesterday, do seeds grow better if they're crowded versus if they have space? Do different seeds germinate at different rates? We could answer those questions using experiment. What's better, coffee or tea? That's not an experiment that we could answer using science. That's an opinion. We can't answer questions of philosophy or opinion or religion using science. So I've given some examples here. These are questions we can answer using experiments. How does the amount of nitrogen fertilizer affect the height of the plant? Does increasing the temperature of an enzyme increase its rate of reaction? Do high doses of vitamin C prevent people from getting the common cold? We can answer all of those questions using some experiments. So that's often the first step. And then we have a hypothesis. We come up with our own hypothesis. And your hypothesis is your prediction. Basically, you set up the experiment and then you predict what's going to happen in that experiment. There's some important things to understand about a hypothesis. So it has to be testable. It has to be something you can actually test in an experiment. Oftentimes we phrase our hypothesis as an if-then statement. If seeds are placed in natural light, then they will germinate at a higher rate than artificial light. So we often will use that kind of pattern for our hypothesis. If this, then that. If people are given twice as much vitamin C, then they will be less likely to get sick from a cold. Often on a Regents exam, they ask you to maybe write a hypothesis. One important thing that they're very picky about, it has to be a statement and it cannot be a question, right? Because your hypothesis is your prediction. You ask a question earlier in the process, but your answer to that question has to be a statement, has to end in a period. Okay. So notice these are all questions, and these are hypotheses that match those questions. These all have a question mark. These all have a period because they're statements.
No way.
So we ask questions. We make predictions about what's going to happen in our experiment, and then we have to actually set up an experiment. So an experiment is just a way of testing a hypothesis. That's what an experiment is.
No, let's go back. Let's go back.
Okay, hold on. So an experiment is a procedure that will test the hypothesis.
Yeah.
And we use something we call controlled experiments, where we're trying to be really careful about what we are doing with our experimental setup. To talk about experiments, let me use an example. Let's use an example of we're gonna use the example of will plants, we're gonna think about the question, will plants grow better if they are fertilized versus unfertilized? This is just sort of a simple example. And probably you're like, of course, they grow better if they're fertilized. Okay, so the question we're asking is that we would come up with our hypothesis. Probably plants will, let's be specific, pea plants will grow taller if they are given fertilizer. That's a hypothesis. Okay, so how would we actually test that? So do I just grow a bunch of plants and give them fertilizer? What's the problem with that? Chelsea?
One has to be tested well, fertilizer, and one has to be tested.
Right. If I just grew a bunch of plants, I gave them all fertilizer, maybe they would grow and I'd say, I don't know, they look taller. But I'm missing something. What Chelsea said. What do we need, Chelsea?
One without it and one with it.
Yeah, we need a group of plants that we don't give any fertilizer, because then we can compare the two. We have names for those groups. The group that we didn't give any fertilizer, we call it the control group. The control group in an experiment is the group where you kind of just give it normal conditions. You're just giving it, you're not treating it any differently. So in this case, it would be the pea plants that are growing with just regular water that doesn't have any fertilizer.
We'll get it for a second.
Can we be quick?
Yes.
That's what we need.
Yes.
Very quick. In fact,
The group that we do
this fast.
give some sort of treatment to, we call the experimental group. The experimental group, we're adjusting something. We're changing something about it. In this case, we're giving the experimental group fertilizer. So a good experiment, you have to have multiple groups, one of which is just treated normally, and one that you're changing something. And so, in all of our experiments that we set up with our seeds, we have multiple groups. We have one group that's in natural light, one is in artificial light. We have one group that is with crowded seeds, and one, the seeds have space. Okay, so we have these different groups. And we also have things in our experiment that we can change. We call these things variables, these factors in our experiment.
Okay.
And so you probably learned this before.
Yeah.
In an experiment, this is something that often students get confused on. So I'll try and give you some ways to help make sure you remember which is which. In an experiment, we have this thing called the independent variable. This is the thing that you are altering. You are changing it to see how it affects something else. In a good experiment, there's only one independent variable. Right? We're trying to change just one thing. And that one thing is what we call the independent variable. Some examples. In that experiment with the fertilizers, I. Yep.
Oh,
I.
So, in our plant growth experiment, whether or not the plants have fertilizer, that's the independent variable. That's the thing that's different. if I'm testing how temperature affects enzymes, I would alter the temperature. That would be the independent variable. If I'm seeing whether or not people get sick if they take extra vitamin C, I would change the amount of vitamin C that people are getting. That would be the independent variable. Let's think about our plant experiments. We have one experiment where the seeds are crowded versus have space, right? That's the independent variable. We have seeds that are growing in artificial light versus natural light. The type of light is the independent variable. So the thing that is different between our groups, that's the independent variable. What I say is the I in independent looks like the number 1 because it's the one thing you're trying to make different between your groups. Right? You only want one thing to be different. The dependent variable in your experiment is the results of your experiment. I tell students, remember, D for dependent goes with the data. It's the data you collect. So what are you going to be looking at to make your conclusion? So in that pea plant experiment, I would probably measure the height of the pea plants, right? I gave some fertilizer, some don't have fertilizer. I'm measuring how tall they are. That's the data I'm collecting. That's my dependent variable. The number of people that get a cold. I'm giving them vitamin C. I'm seeing how many get a cold. How many students get a cold would be my dependent variable. That's the data I'm collecting. What is the dependent variable in our seed experiments? Anyone have an idea about that? What data are we collecting that's going to help us? Chelsea?
The growth.
Yes, like some people are measuring are we all measuring like how tall they got? Yeah. So the height of the plants. We could also measure things like what percent of the seeds germinated or not. Can measure like how long is the root that starts to grow. Yeah, those would be examples of the dependent variable. And then finally, we have these other variables. And I talked to some of you about this as you were coming up with your experiment. If we only want one independent variable, we only want one thing different, there must be a whole bunch of things we're trying to keep the same. So if I have my plants and I'm fertilizing some and not fertilizing others, I want everything else to be the same. I want them to get the same amount of water, the same type of soil, the same number of seeds, the same amount of light. So to really see, is it how the fertilizer is affecting these plants? I need to make sure that everything else is the same. Those things that we try to keep the same, we call them the variables that are held constant. Sometimes we say controlled variables. These are factors we keep the same between our groups. So usually there's lots of them. In the cold experiment in vitamin C, I would want the people to be like the same age, probably. Right? And same health and have the same diet. Because I just want to know how vitamin C is affecting them. In our seed germination experiment, right, we want all of our seeds in the same type of container. We wanted to all use the same type of soil. We want them to be at the same temperature. So we're doing all of that because we're trying to make sure that only one thing is different, and that's the independent variable.
Also, are you recording? It appears in
Okay, so we have our experiment. So we asked the question.
So are you recording this?
Yes. We came up with a hypothesis.
This is gonna sound awkward.
We set up an experiment using all of those rules we just mentioned. And then the experiment's going on. After our experiment is over with, or while it's going on, we make some observations. What that means is we collect data from our experiment. As we just said, we are going to be measuring the growth of the plants. That's our dependent variable. You measure the dependent variable. Sometimes just once at the end of your experiment, other times while the experiment's going on. Sometimes we're recording like things with a number. We call that quantitative data. Like the height of the pea plants. That has a number, that's quantitative. Other times it may be what we call qualitative, based on the color or appearance of things, might be examples of that. And we often will try to organize our information in a data table, okay, or use a graph to sort of show our data in a way that's easily understandable. So for the pea plants, we would maybe measure their height every 3 days. That would be one way that we would collect data for that experiment. We might survey people to see how many people got a cold after having extra vitamin C. For your seeds, it might be how tall the plants got. Those are examples of data we would collect. And then finally, we look at that data. We look at our results, and we try to analyze and see, okay, is my hypothesis correct? I thought this would happen. I've now collected data. Was I correct or was I not correct? Is my hypothesis supported, or does the data not support my hypothesis? So as an example, my pea plants. I might collect my data if the fertilized pea plants were 8.3 centimeters taller than the control plants. That would support my hypothesis. If Ray in his seed experiment finds that the plants in the window germinated and grew much taller than the plants that are in the growth chamber, that would support his hypothesis. So the data is what you use to come to a conclusion about whether or not your hypothesis was correct. So sometimes your hypothesis is supported by the data. So what do? Well, sometimes you might repeat the experiment again to see and confirm the results. You might make sure you have all your variables controlled. Make sure you have what we call a valid experiment. And what scientists do is they actually share their results with other scientists. The name for that is peer review. What scientists do after they've completed an experiment and found out some information is they share it with other scientists. They actually publish it in these sort of magazine kind of things. And other scientists subscribe to these. They're called journals. And other scientists subscribe to them, and they will read through them to see, okay, what are other people that are interested in the same thing that I'm interested in? What are they finding? What is their research showing? Wait. Sometimes, though, our data doesn't support our hypothesis. Sometimes we collect our data on our dependent variable, and what we thought before the experiment is not supported by the data. And that's okay. Our hypothesis, what we say is our hypothesis was rejected. The results don't support what I thought earlier. That's okay. It can lead to new investigations, new ideas. We still learn something from that hypothesis, even if it's rejected. So when we design experiments, it's important that the experiments are what we call valid. That word, it means basically like reliable, that they were correctly designed. And one of the important things, and I talked to some of the groups about this yesterday, is you want to have typically a large number of samples. You don't just want like one seed in each condition. You want multiple seeds. And the more data you have, the more samples, the more confident you can be in what you find, because it's been shown with multiple samples. We also want to be sure we're controlling as many variables as possible so that we only have one independent variable. All right, so I'm going to have you do something here for a minute. So let's imagine this scenario. I know it's similar to one we've been talking about, but an experiment's conducted with 30 pea plants. 10 of them are given no fertilizer. 10 are given 5 grams of fertilizer. And 10 plants are given 15 grams of fertilizer. And after 2 weeks, we measure the height of those plants. On your slide, you have a few questions. What's the hypothesis? What's the independent variable? I'd like you to take a few minutes. If you need to look back and remind yourself of what those terms mean, go ahead and go for it. I'd like you to answer those questions about this scenario with the pea plants. And then we'll review them to sort of make sure we're on track and understand some of these terms. So go ahead and answer those questions, please. All right, so let's think about this experiment. So we have 3 groups of plants. One group got 0 fertilizer. One group got 5.
Yes.
One group got 15. We measure their height at the end. So a hypothesis in this experiment that we're testing might be the plants given more fertilizer will grow taller. It's your prediction. A couple of people had a question here. It can't be a question. It has to be a statement. So that would be a hypothesis. The independent variable. This is the thing that's different between these 3 groups. The one thing you're trying to make different, in this case, it's the amount of fertilizer. That's what's different between these 3 groups. They're getting each different amounts of fertilizer. The dependent variable, this is the data you're collecting. the information you're gathering that's going to help you make your conclusion. In this case, it's the height of the plants. That's what you're going to be measuring. If we're setting up a good experiment, we want them to get different amounts of fertilizer. Everything else should be the same. The type of soil, the amount of light that they're getting, the amount of water. You can come up with a long list of those. It doesn't have to be these two. The control group, this is the group where it's just treated normally. It's not getting anything changed about it. In this case, it's the plants that got no fertilizer. We're going to be using those as a comparison. The other two groups that are getting fertilizer, those would be the experimental groups, the one that got 5 and the one that got 15 grams of fertilizer. And how can we improve an experiment? You could always improve an experiment by using more samples, using more people. Rather than just 3 groups of 10, maybe we have 3 groups of 50. That would be a better experiment. That would be more valid. So we'll continue practicing with this tomorrow. But that's where we'll end for today.